All right, this last section we'll look at proteins and nucleic for chapter 2. All right, so let's start out with proteins and amino acids. So amino acids, these have an amino group, a carboxyl group, and some type of functional group. All right, so if we look in this image, here we have our amino group, and amino means that it has some type of nitrogen. Then we have our carboxyl group, C double bonded O, and then 2 and OH. And then this R represents some type of different functional group. So there are 20 different types of amino acids that we find within the body. And so amino acids are going to be charged, and so they'll attract one another to form kinks and folds when they make different types of proteins. And the functional group is what differentiates then the 20 different types of amino acids. So examples of amino acids then are arginine, aspartic acid, tyrosine, valine, cytosine, and so the sequence of amino acids then in a chain is determined by its DNA. And when we have several amino acids, then what we get into are proteins. And so then the function of both proteins and amino acids then are structural, where we have collagen fibers and connective tissue. Also, we have keratin within our skin to help with that protective barrier. Then we have enzymes, which these assist every chemical process within the body. We also have antibodies. These are part of the immune system and help with the immune response. We have different types of receptors, which are found within the cell membrane and also intracellularly, so that cells can talk to one another and communicate. And then also we have carriers, which are found across the cell membrane or in blood, so that they can transport different ions and molecules across. When amino acids are joined, a hydrogen is stripped from the amino end, and then so here is in our example, so amino end again has to do with nitrogen, and then an AOH or hydroxyl is stripped away from the carboxyl end. So here, on the carboxyl end, we have OH being removed. And so this forms a peptide bond. All right, so my question for you is, since water is going to be one of the byproducts when we make this peptide bond, what type of reaction is this? So if you said dehydration synthesis, you're exactly right, because one of the byproducts is we're removing water and we are going from small molecules into larger molecules, all right? And so when amino acids are added together to form a chain, this is called the primary, or one with the little dot is also primary structure of a protein. From those chains of amino acids and that primary structure, in this example here, okay, so the chain of amino acids, this is called, what we have is a polypeptide strand or polypeptide chain. Multiple amounts of amino acids that have been formed together by dehydration synthesis, to reiterate. So the chains can vary in length from 3 to 4,500 amino acids. And then what we go on to then are the secondary structures. And there's two different types. So we have the alpha helix structure, and then we also have the beta pleated sheets, both of which though are going to be formed by hydrogen bonds. Okay, and this may form between neighboring amino acids based on their different charges. So when they have this helical arrangement in space, this is called the alpha helix, versus these sheets, which are going to be these pleated sheets. Okay. These beta sheets typically act as precursors and tend to be very small in size, like 3 to 10 amino acids long. But more often, if there's a high accumulation, these tend to have different pathological conditions and have been like associated with Alzheimer's and like neurofibrillary tangles. More common, what we have are the secondary structure of the alpha helix. And so after with the alpha helix, then we'll have more hydrogen bonds that are going to create more bends and folds and create now a 3D spatial arrangement that we have. And so those helical structures are just going to bend and fold on themselves. And so this is now called the tertiary structure. And this is where now structure will dictate its function. Weak bonds, hydrogen bonds, are going to hold the structure together. And proteins are easily denatured here, and then by like changes in temperature or pH. And this is where we get a vast majority of our proteins. So if you've ever fried an egg before, what you're doing then is you're denaturing or unfolding the proteins that are found within that egg. And so that's why it goes from that clear structure and then to that, or clear color to that white color. And then the last type of protein structure that we have are the quaternary structure. And so some functional proteins are composed of multiple polypeptide chains that now are covalently bonded together. And so examples of this include hemoglobin, where we have 4 polypeptide chains in blood, and insulin would be another example where it has 2 polypeptide bonds with it. Conjugated proteins occur when there are proteins that are combined with other molecules other than proteins to become functional. First example we'll look at are glycoproteins, and so glyco means the carbohydrate and then protein for protein. We'll see this with a lot of hormones and especially within the cell membrane for cellular recognition and for communication of self. Another example are lipoproteins, and so lipo would represent a lipid, and then as well as now the protein portion. And we find lipoproteins within cell membranes and also different carrier molecules within the blood. When we look at this table here are other examples that we see of other molecules with proteins. And so here we have the protein and then what the non-protein component is. So for hemoglobin, we're adding some type of heme pigment, and so this is important for carrying oxygen within the blood. Myoglobin has also a heme pigment, but then this stores oxygen in muscles, very similar in action, just a little bit different to hemoglobin. Insulin, there has additional genes, and so that's important for hormonal regulation. And then we also have blood group proteins like albumins, and so these also have carbohydrates which help to produce different blood types, so like our ABO blood type. And then lipoproteins, which have lipids and then help to transport lipids within the blood. Next, going on to now DNA and nucleotides. So nucleotides are going to be the building blocks for nucleic acids. And so these will have the same type of composition. So they have a 5-carbon sugar, they'll have some type of phosphate group, and then some type of nitrogenous base. And it's the nitrogenous base that will determine what type of nucleotide it is. So the nitrogenous base falls into 2 different categories. We have pyrimidines and purines. Pyrimidines have only 1 carbon ring and a nitrogen, and so these include cytosine, thymine, and uracil. And then purines then have 2 carbon rings and a nitrogen, and so these are only of guanine and adenine. And then the pentose sugars are either going to be deoxyribose or ribose. And so for the deoxyribose, this is for DNA. And then for the ribose groups, this is for RNA. And so that's what's going to make the difference between the two. With DNA, or deoxyribonucleic acid, the deoxy means that there is a lack of an oxygen molecule within that sugar ring. And so the sugar in the molecule is called deoxyribose and can bind to one of 4 nitrogenous bases. Each of the nitrogenous bases form a hydrogen bond with another to result in a double-stranded molecule that we see here. Okay, and so here we have the pairing, for instance, with adenine and then with thymine. And then we have guanine always pairing with cytosine. So with DNA, C's will always pair with G's, and then T's will always pair with A's, okay? So C and T cannot pair together, and G and A cannot pair together, or this and this. That will not work at all, okay? So it's only between this guanine and cytosine, and then thymine and adenine in terms of the base pairing. So then how we get this helical structure then will be due to hydrogen bonds. And so these are relatively weak in terms of strength. However, so DNA is going to be then the basis of the genetic code, and then the sequence of bases codes for amino acids then to make proteins. So then the formation of proteins then goes into RNA or ribonucleic acids. And so the difference with this now is that we have an oxygen molecule in that sugar ring, so that's why it's the ribonucleic acid instead of deoxyribonucleic acid with DNA. And so same concept here, the only difference is that instead of thymine that we saw with DNA, what we now have is uracil. And so if you look at the structural difference, we're missing a methyl group, and so now instead it's just a hydrogen group. So RNA is a little bit different too in terms that it's a single strand instead of a double strand when we have this. So there are 3 different types of RNA, and so these are used to make different types of information for assembling a protein after it has come out of the nucleus in DNA. And so to actually assemble it, then we have these 3 different types. We have mRNA, tRNA, and rRNA. mRNA stands for messenger RNA, and so this is a specific amino acid sequence of protein products of gene expression. tRNA is transfer RNA, and it carries amino acids to mRNA at the ribosomes. And then rRNA, ribosomal RNA, is essential for protein synthesis and is what forms enzymes.